and Lösante. Welcome. Lösante Hospital is full of excitement today. We feel greatly privileged and honored to welcome you to Lösante Lösev inaugural International Cancer and Life Symposium, which we hope to be an annual tradition. Thank you for being here to share our pride, joy, and excitement. Cancer, an unquestionably unwelcome experience in anyone's life. We have many guests here today who know firsthand how it feels when it happens to a child. Dr. Üstün Ezer, a pediatric hematologist-oncologist, felt this pain and despair deep in his heart 24 years ago and went ahead and took the steps with a small group of colleagues and fathers of children with leukemia to found Lösev, Foundation for Children with Leukemia. With limited resources, but unlimited hopes, dreams, and ambition, and a powerful motto: there is nothing more valuable and important than a child's life. And here we are today, at this world-class 400-bed hospital. Thousands of treated children with a greater than 92% success rate. A great, unique model of integrative approach of treating the whole child, from psychosocial support to food programs to education. Please join me in welcoming the physician who initiated it all, the founder and president of Lösev and Lösante Hospital, Dr. Üstün Ezer, for his opening speech.
Good morning. My English is not perfect, so if you let, I'll speak Turkish. I'm sorry. Evet sevgili konuklar, Türkçe konuşmayı daha kendimi rahat ifade edebildiğim ve kendi olduğum için tercih ettim. Hepinize günaydın, değerli konuklarımız, değerli bilim insanlarımız, değerli arkadaşlarımız. Hakikaten bugün heyecan verici ve tarihi bir gün olarak algılıyorum. Ben de heyecanlıyım. Hepimiz için güzel bir kongre olmasını, sempozyum olmasını ve daha güzel günlere, daha bilimin ağırlıklı olduğu toplantılara vesile olmasını diliyorum. Biliyorsunuz 90'lı yıllarda ben pediatrik hematoloji ihtisasını yaptıktan sonra mesleğime başladığımda hemen hemen birçok hastamızı, lösemili çocuğumuzu ne yazık ki kaybediyorduk. Ve bu kayıpların altında en büyük sebepler enfeksiyon, kanama, hijyenik ortamların sağlanamaması ve maddi sıkıntılar, maddi yokluklardı. O yıllarda en iyi ihtimalle %20'lerde olan tedavi başarımız 90'lı yıllarda ne yazık ki beni mesleğinde bir yol ayrımına getirmişti. Ya bu işi başka bir şekilde düzeltmek zorundaydım ya da bu mesleği farklı bir alanda yürütmek zorundaydım. Yani bir yerde mesleğimi yapmama, bırakma noktasına gelmişti. Çünkü çok fazla çocuk kucağımızda ölüyordu ve çaresi de yoktu. İşte 1998 yılında arkadaşlarımla birlikte gelmemiş miydik daha önce? ...1998 yılında bir grup arkadaşımla birlikte Lösev'i kurduk ve lösemi tedavisindeki başarıları nasıl arttırırız diye el ele verdik ve büyük bir çaba sarf ettik. Hem doktorluk yapmak, bilimsel gelişmeleri takip etmek, hem tedavilerdeki hataları veya uygulamalardaki hastane ortamlarındaki hataları, eksikleri gidermek için gerçekten insanüstü bir çaba sarf ettik. Bir de şunu görüyordum ki sadece ilaçla, sadece tıbbi olanaklarla tedavilerdeki başarı yüzdemizi, survey'imizi yükseltme imkanı yoktu. Bunun yanı sıra çok iyi beslenmeleri gerekiyordu hastalarımızın. Hijyenik ortamlarda yaşamaları gerekiyordu. Enfeksiyondan korunmaları gerekiyordu. Ama en önemlisi de psikolojik olarak büyük destekler almaları ve hayata sımsıkı bağlanmaları gerekiyordu. Çünkü umudu olmayan bir hastanın hangi ilacı, hangi tedaviyi verirseniz verin, umudu olmayan bir çocuğun iyileşme şansı çok azdı, çok düşüktü. Bunun çok örneklerini ben gördüm, yaşadım arkadaşlarımla birlikte ve tedavi yaklaşımlarına hem sosyal hem psikolojik hem bir yerde de tabii Türkiye'de çok büyük sorun olan maddi katkılarla destek verdik ve bu desteklerle bugün hem ulusal hem uluslararası yayınladığımız üzere %92 3 yıllık survey'i %92 başarı elde ettik. Tabii kemik iliği transplantasyonunun başlamasıyla da hastanemizde, bu ortamda, bu binada çok daha iyi koşullarda %2 de transplantasyonun getirdiği yaşama katkısı, survey'e etkisi oldu. Bugün gururla açıklayabiliriz ki yine uluslararası bütün bilimsel makalelerde veya dergilerde veya kongrelerde açıklayacağımız üzere %94 gibi bir başarıya ulaşmış durumdayız. Tabii bütün bunları bilimsel ortamda yapmanın ve bütün uluslararası kuruluşlarla, değerli bilim insanlarıyla paylaşmanın da ayrı bir önemi ve gereği var. Ben bu binanın projesinde mimarla birlikte çalışırken, bu hastanenin projesinde mimarla birlikte çalışırken böyle bir salonu kendisine söylediğimde, hocam ne yapacaksınız bu kadar büyük salon, bir özel hastanede böyle bir salon ne gerekir demişti. Benim vizyonumda, benim hayalimde burasının tabii ki üniversite olması, bir an önce tıp fakültesi olması ve birçok bilimsel kongreye eşlik etmesi, zemin hazırlaması vardı. Bugün bu heyecanı hakikaten doyasıya yaşıyorum. Çünkü çok değerli bilim insanları dünyanın çeşitli yerlerinden, ülkelerinden ve kendilerini böyle hem yazılarında hem çalışmalarında izlediğimiz ve gururla örnek aldığımız bu bilim insanlarının aramızda olması gerçekten gurur verici bir şey. Ama tabii bu sempozyum bir giriş, bir prova niteliğinde oldu. Esas bizim büyük sempozyumlarımız, kongrelerimiz 25. yılımızda, vakfımızın kuruluşunun 25. yılında önümüzdeki sene olacaktır. 2023 yılında çok daha büyük kongrelerde inşallah hep birlikte bu çatının altında, bu bilimsel yuvada olmanın gururunu yaşayacağız. Ben bu arada kongrenin düzenlenmesinde veya sempozyumun düzenlenmesinde kısacık bir süre içerisinde emeği geçen bütün arkadaşlarımı kutlamak istiyorum ve tebrik ediyorum kendilerine. Güzel bir sempozyum olmasını, katılımcı bilim insanlarına özellikle büyük katkılarında bulunmasını diliyorum. Hepinize katılımlarınız için çok teşekkür ediyorum. Misafirlerimize, konuklarımıza da geldikleri için, bizlere destek oldukları için çok teşekkür ediyorum. Umarım daha güzel bir ortamda, daha geniş, daha büyük kongrelerde bir arada oluruz. Hepinize çok teşekkür ederim. İyi günler diliyorum.
Thank you, Dr. Ezer, for such an inspirational speech. Our next speaker, for his opening remarks, is an exceptional leader in pediatrics, Professor Dr. Enver Hasanoğlu, President of International Pediatric Association, IPA, a global body representing the professional societies of pediatricians connecting more than 2 million pediatricians from all over the world. Professor Hasanoğlu is also the Secretary General of the Union of National Pediatric Societies of Turkish Republics and Turkish National Pediatric Society. He is also a National Committee member of Directory Board of UNICEF Turkey and has served as the President of Gazi University and as a member of Turkish Higher Education Council. Please welcome Professor Dr. Enver Hasanoğlu for his speech.
United, dear colleagues and attendees, I would like to welcome all of you to this symposium. As the President of IPA, International Pediatric Association, and Executive Director of Turkish National Pediatric Society, of course I am going to speak about children and their problems. IPA, it means International Pediatric Association, was founded in Paris by a group of European pediatricians in 1910. Today, with 149 national, 7 regional, and 10 subspecialties societies working under umbrella of IPA. IPA is trying to improve the well-being and health of 2.5 billion of children within the world with 2 million of pediatricians. Our mission is to reach every child, every age, everywhere. And we are working closely and collaborating with WHO and UNICEF to make our children more happier and healthy and well educated. Nowadays, we are facing, and our children facing, some problems. I'm going to focus on this. First of all, as you know, it is COVID-19. About 3.5 million children we have lost it during this pandemic. And of course, so many of them still suffering from the complication of the pandemic, as depression and anxiety. Also, with the school being closed, they got affected psychologically and physically, and the ratio of obesity began to increase. And to closer of the schools, the early marriage among the girls began to increase. Secondly, climate and environmental change, which cause drought and scarcity of clean water, and thus the communicable diseases began to increase. So the death of children began to increase as well. The third big problem is vaccine hesitancy or refusal of vaccine. This problem increased during COVID-19 pandemic, and thus the rate of preventable infection diseases like measles began to increase as well. Fourthly, the problem is the refugee problem. About 1% of the globe population are refugees or looking for asylum, or they are replaced from their country. And the number of children around the world is about 3 and 2 million children are refugees or looking for asylum or replaced. The majority of these refugees, as you know, from Syria, Yemen, Afghanistan, Venezuela, Sudan, and Myanmar. Turkey is hosting more than 5 million of refugees in our country. And without any doubt, after this conflict in Ukraine, the number of refugees will increase. IP is trying to reach children suffering from non-communicable diseases as well, like cardiovascular diseases, obesity, diabetes, and Lösante malignancy. These non-communicable diseases, as you know, related to genetics, psychology, and environment change. More than 4 million people die annually from these diseases. 70% of the death is related to non-communicable diseases. And 20% of this ratio is in children, and their age is between 10 and 19. The malignancy ratio in children is about 2.5 percentile, and about 300,000 children under the age of 15 suffering from malignancy. SIOP, it means International Society of Oncology, Pediatric Oncology, is our subspecialty society. We are working with them, collaborating with WHO, and trying to fight against this malignancy. I think the next meeting of SIOP will be in Barcelona next September. They will discuss the malignancy program in details there. If I come to Lösev, Lösev is non-governmental, non-profit organization trying to reach the children whom suffer from malignancy and leukemia, and treating the patient free of charge. Lösev achieved great success in their action and treatments. Turkish National Pediatric Society and International Pediatric Society appreciate this success. I'm sure you will discuss during these 2 days about the malignancy in our children in great details. I would like to thank all of you for being with us, and thanks to our speakers, and of course to organizing committee, especially to Professor Emin Kürekçi. I'm sure this meeting will terminate as successful as it began. Thank you so much and good luck.
Thank you, Professor Hasanoğlu. Now, we will welcome the Symposium President, Professor Dr. Emin Kürekçi. Professor Kürekçi is the Director of Pediatric Hematology Oncology and the Director of Pediatric Stem Cell Transplant Center at Lösante Hospital. He is also a member of the Board of Directors of Lösev and Lösante Hospital. Priorly, Professor Kürekçi has been instrumental in starting the pediatric bone marrow transplant program at Gülhane Military Medical Academy, where he also served as the Director of Pediatric Hematology. It's my honor to invite Professor Dr. Kürekçi on the stage.
Thank you, Işıl. And good morning, all. Dear colleagues and distinguished guests, this year, as we celebrate the 21st Lösev International Month for Children with Leukemia, we are proud to organize and host the inaugural International Cancer and Life Symposium at Lösante Hospital. Cancer is a major public health problem in Turkey with more than 200,000 new cases each year, unfortunately. Lösev as a foundation has taken upon itself not only free medical services for cancer patients, but also education, employment creation, and research as its mission. Lösev was founded 24 years ago in order to fight against childhood and adult cancers. And now it is currently a national leader among non-profit organizations in our country. We are very proud of it. An important part of our education mission is to make sure that our children undergoing chemotherapy are not falling behind their peers. For this purpose, we established a particular program in Lösev schools. I would like to thank their dedicated teachers who provide the highest quality education to children with cancer at no cost. We are also getting ready to transform into a higher education institution by establishing a health sciences university, namely Lösev University. Today, we have come together here to join forces with science, knowledge, and experience in the fight against cancer. I would like to offer my deep gratitude to all the guest speakers and session chairs for their contribution to the symposium with their talks on very intriguing hot topics and providing the light of hope to the lives of those living with cancer. I also would like to thank you for your participation. I hope to meet again on many more cancer and life symposiums. Thank you.
Thank you, Honorary Presidents and Symposium President. I'm sure, just like me, you can't wait to hear from our distinguished guest speakers. It's time to start the first session of today on childhood leukemia. May I invite session chairs, Professor Dr. Ülker Koçak from Gazi University Faculty of Medicine, Division of Pediatric Hematology, and Professor Dr. Şule Ünal Cangül from Hacettepe University Faculty of Medicine, Division of Pediatric Hematology.
Thank you, and welcome to the scientific session, dear colleagues and dear guests. We have two very distinguished and famous speakers in this session. I don't think that they need any introduction, but I have to say that I have first met Dr. Andrea Biondi when I was a resident in 1995, and he's from Milan, Italy, and not only me, but all the leukemia treaters are very excitedly following his work on molecular biology on childhood ALL, and so we will be very happy, and it's my honor to introduce Dr. Biondi for a speech on how he treats relapsed and refractory childhood ALL. Thank you.
Good morning. First of all, many thanks for your words, and I'm really honored to be here today. After reading the booklet that I received about this foundation, I was very impressed, and this is a special honor for me to be here and to share some thought about the future of the relapsing ALL, and later on about the genomic of ALL. I am fully aware that when we are talking about this pediatric cancer, we need to be aware that still the success is dealing with a minority of children around the world. And overall, I think it's not more than 20%. And this is a strong commitment that, as the president of IPA said, for all of us as a pediatrician, to think that it's not only the success in a limited population, but to have a broader view. And in that sense, I think it's very important what the president of IPA said about this, the important commitment that even the WHO took with respect to this childhood cancer initiative in order to bring the perspective of the children improvement around the world. How we treat relapsed refractory ALL at the present time and the future perspective. First of all, let me just give in 2 or 3 slides what are the background of that, to see where are we, and then try to foresee and discuss what are the challenges that we are facing today in order to further improve what is still the cancer with a high prevalence, because relapsing children for ALL is still the top, the first in the list of the pediatric cancer in terms of incidence. And this is just to highlight what are basically the concept. The prognosis of relapsing ALL is gradually dependent by time of diagnosis to relapse, the site of relapse, and the type of immunology, and later on we'll see the genetic of this disease. And this is basically the background. I have to acknowledge what has been done by the Berlin Frankfurt Münster study group, that probably was one of the first that highlighted the possibility to successfully treat the children who relapse, who are resistant, by, let's say, standard second round of chemotherapy and transplantation. And but just to look at this type of schema, which have become the standard of classification of relapsing ALL, you can easily highlight what are the challenges, because we are still not curing all the kids, as you can see from the first column. And the other important thing is that we still need to reach this goal to use the bone marrow transplantation, which is an essential component of our treatment. But unfortunately, if you look prospectively and you consider the price that you have to pay when you need to perform bone marrow transplantation in a child, and you need to perform a total body irradiation, this is something that, of course, if you look prospectively, you want to reduce as much as you can. Even before, as a pediatric oncology, I did honestly expect that the results of this fantastic job that the colleague from Vienna, Christina Peters, brought together. I have to say that we as pediatric oncologists, we must be proud of the fact that we take the challenge of very difficult questions and perform what are the right study to give the right answer, which are basically the randomization and randomized study. And Christina Peters did the job. Can you imagine to convince bone marrow transplanter to accept a different type of conditioning regimen? Very challenging. But she did. And unfortunately, I have to say, the results clearly show that the TBI is still superior for any type of subgroup. In that sense, I'm not happy about the results, because I did expect to be the opposite. In order to get a conditioning regimen that preserve the fertility, the height problem, and the growth in a cured patient. The other things that I want to share with you that we are challenging today with respect to the biology of relapse is also a sort of change in the paradigm from the classic model that following Darwinian concept, you would expect that you have why do a patient relapse? Or simply because you have a progressively acquisition of new mutation, and accordingly selection of clone resistant to therapy, and then of course relapse. But this type of paradigm is today challenged by the fact that it's likely that probably relapse-fated clones are probably already present at diagnosis before any treatment, with the specific biological features, which are basically chemo resistance, they are quiescent as a stem cell, and the lack of specific genetic alteration. This clone can survive to the treatment and then induce a relapse. And of course, if you look prospectively, you say, well, the best things would be just to identify those cells and try to kill before getting the relapse. And if I had to see prospectively, this is going to be probably possible in the future, because the single cell RNA-seq is becoming something not available routinely, but can definitely provide some insight of the heterogeneity of, at the clonal level of the bulky disease that we have a diagnosis. So if that is the scenario that we are facing, what are the challenges in that perspective? And instead of taking just a general perspective, I took what is under discussion within the international BFM, that, as you know, is the European Consortium of Leukemia Lymphoma, and which basically gathered together most European countries, and not only beyond Europe as well. And this is just the ongoing discussion of the design of what is called the inter-real, so relapsing ALL strategy of the PCP within this group, which cover basically all European countries, in order to highlight to you what are the changes that we are facing with and actually whether they're going to be better in terms of results, we still don't know, but clearly provide a landscape of the scenario. First of all, the definition of risk is becoming more precise by including, as you can see, a new category of what is called the very high-risk leukemia. And the very high-risk leukemia are those who are carrying specific genetic alteration and are very relapsed early in time, below the 18 months, so basically during treatment. Then there is a standard risk as defined and the high risk. And what is the key first message that I'm sharing with you? That the major breakthrough at the present time in our field, not only in leukemia lymphoma, is coming from the immunotherapy and immunological approaches, which are basically under investigation in childhood level. I have to say that anytime I'm presenting this slide, I am fully aware on the issue of the access to this type of strategy around the world. But I am an optimistic, and a pediatric oncologist cannot be an optimistic person. And I believe that we should, to some extent, think about that even the cost of drugs should become more available around the world. Because if something is useful for a child, that should become available and not just be restricted to the wealthy countries. Why immunotherapy is an important breakthrough? Simply because it's likely that can provide the same type of efficacy, but with a less side effect. That's still to be proved, you can say. But definitely if you consider as a paradigm as compared to chemotherapy, where of course you are providing intensive chemotherapy with all the consequences, especially the late effects, definitely immunological approach is one way. And I would like just to highlight three major themes which are currently in this field for relapsing ALL: the monoclonal antibody and the manipulation of the immune system with the CAR-T, to explain why from this type of approach we will have in the future. I'm old enough, maybe your generation will see the benefits of that. And the first one is blinatumomab. Blina is an antibody which is a very fancy. When you think about them, talking to the young people, the person really think about this generation. It was a German colleague that basically think about the possibility to create artificially a sort of synapses between the leukemia cell and the immune system, the T cells, just by building up an artificial molecules which recognize the antigen on the leukemia, and the other side as a synapses on T cells, the CD3. This is basically blinatumomab. And how does it work? Just bringing together the leukemia and the T cells. Why I'm saying that? Because two independent studies, one in Europe and another one in US, brought the same conclusion. Then in the treatment schedule of a patient who relapses, depending on the type of relapse, according to the classification, but in both cases they observe the same results. If you include 1 or 2 cycles of blinatumomab instead of the heavy intensive chemotherapy before transplantation, you can achieve better results. Can you imagine? Just by changing 1 cycle of intensive chemotherapy or 2 cycles. And this, of course, is something very important, because bringing a patient to bone marrow transplantation in a better shape with the low incidence of risk of relapse of infectious disease is definitely better. And this has been shown, I have to say, surprising, because if you look to this study, you see, look at the type of study that was both randomized, 2 blina cycle versus 2 chemotherapy, or 2 chemotherapy, and then just a change of 1 cycle. And the results were dramatically clear, as it happened when a randomized study is clearly well designed. Look at the arms of the Blina versus chemotherapy. All those patients were transplanted after that. So it was just a way to conditioning before the transplantation in order to achieve the best remission that you can achieve without, but with less intensive treatment. And this is basically the results of the American study. You see exactly the same, suggesting that now, if you have to transplant a patient after resistance or relapse LL, of course, it would be important to use Blina instead of chemotherapy cycle. But then is a question of the cost about that, which I think is, I'm always aware about that, and we need to think about some solution in order to get, at least for children, access to a lower price drugs when it becomes relevant for them. And this is a clear relevant condition. Inotuzumab, another interesting monoclonal antibody, completely different type of molecules. You have the antibody, and at the tail you have a toxin. And only when the antibody recognizes the antigen on the surface of the cell, you have the process which is called internalization, and accordingly, when the antibody is inside the cell, that you have the cleavage of the toxin and accordingly the effect. Why it is important? Because as you have seen in the schema of the working strategy for LL, we are going to test whether to a child who relapses, especially to the very high risk and high risk, instead of providing another cycle of chemotherapy. I'm always very puzzled by the fact, and I'm old enough to say that, that after intensive frontline chemotherapy, I have to say, although it's been successful, it's not logical to provide another cycle of intensive chemotherapy. You need to find alternative strategy to achieve remission and to bring the child to transplantation, if needed. Because I have a great hope, and I hope not to offend any transplanter in the room, that even for this type of disease, in the future, we can probably cure without transplantation, for the reason I explained before, that at least for ALL, we still need to perform TBI with all this light effects. The other important breakthrough, which is relevant for relapsing refractory, it came from this fantastic new page of medicine, the CAR-T. Can you imagine, for the first time, we are all used to call the pharmacy for a drug. But this is a special drug, because you just harvest the lymphocyte from the patient, then you send to the company sends you back the lymphocyte which has been manipulated genetically to become a drug. This is the first time in my life, except for transplantation. In other words, the cells of the patient are becoming the drug for the patient itself. And of course, this is just the product which has been approved by the FDA and by the EMA for the treatment of relapsed refractory LL after 2 rounds of chemotherapy treatment in the persistent relapse. And it is called with a very fancy name. I don't want to pronounce. Let's call Tisagen. That's probably easier. And you see where the structure came from. Again, it's a novel molecule. It does not exist in nature, because it's just the results of the genetic engineering bringing together the concept of the antigen binding domain from an antibody to the just a portion, which is a transmembrane portion. But what is very important, you see in the tail, the fact that you need to have the coding sequence of what is defined a co-stimulatory molecules. What are co-stimulatory molecules? Are the molecules which are essential in our immune response to provide the signal to the lymphocyte to proliferate in order to keep the infection under control, but then of course to stop it. Otherwise, every time you have an infection, you will have an explosion of your lymphocytes. So these are the essential components, and these are basically a CAR. And according to the different type of co-stimulatory molecules, you can define generation. Why this type of approaches is relevant? Because when you have a patient with a resistant refractory disease, even after 1 or 2 transplants, what are you going to expect from the life of the child? Almost nothing. We have no treatment that can give any chance to the patient to survive. But if you see in the bottom part of this cartoon, well, of course, you have the experience of the clinical trials, the real world, and even our experience in Monza, which has been the largest center in Italy with the experience of this CAR-T technology, together with Rome Bambino Gesù hospital in Rome, as I said. Approximately 1/3, 30% of the patients who have a disease with no chance of being treated can achieve a long-term remission. And you can argue, well, 30% is not so high. But you have to consider that those children do have any chance, no chance to be treated with the current protocol. Consider a child with ALL who relapse, first bone marrow transplantation, might be a second one, and then later on relapse. What you can expect from this situation. And that is unfortunately the real situation. But today we have potentially a new tool, even to treat that very bad cases. Unfortunately, this type of treatment has a very important safety issue because, not surprising, when you have an explosion of immune reaction, what do you have in your body? A release of a storm of cytokines. What is fever? Fever is just the result of the cytokine release, of the consequence between the chain reaction between lymphocytes and the target. So according to that, the question is, it's a fantastic tool, but we are still at the beginning. And why we are still at the beginning? For the reason which I've listed. First of all, for the complexity to have this type, access to this type of drugs. Again, you are not ordering to the pharmacy. You have to harvest the lymphocyte by an apheresis, send to the company, get it back, and then infuse. And you can consider what is going to happen in the meantime. Might be that you have an explosion of the disease, and accordingly, the patient dies, which is different from having the drugs directly from your pharmacy. The toxicity, surprising, more than 50% of relapsing after CAR-T, leukemia have lost the antigen. I was very impressed by that. Even if you think about the Darwinian selection, it's very challenging, that you are losing the target of your intervention. And many other issues, for example, failure to collect T cells. So let's see rapidly what are we going prospectively to think about on how to overcome this type of approach that I have no doubt will become in the future a strategy not only for treating acute lymphoblastic leukemia, relapsing refractory, but even earlier on. It's fantastic to think about that your immune system could become, from yourself or maybe from a donor, the way to treat in a better way, more targeting your disease, instead of using very aggressive chemotherapy or even bone marrow transplantation, including TBI? Do we have alternative to produce custom-made? Yes, but keep in mind that at this time, all CAR-T has been recognized as being a drug, and accordingly should fulfill all the process of becoming a drug and approval. But there are an active research to think about why, for example, by using this machine, you can produce in your setting. Of course, you need to have all the requirements of the reagents that should be approved under GMP, general condition for getting a drug infused in a patient. We need definitely to reduce toxicity, yes, because it's experience of all of us who have been treated children with CAR-T that sometimes you need the ICU for these children. And we are not surprised at pediatric oncology of having a child in an ICU, but this is part of the process because it could happen frequently. And of course, there are many strategies. We need to have a CAR-T more effective, because if you have a relapse of a disease which has lost the target of your intervention, that implies that you need to have more effective. And look at how many strategies are currently investigated in many places around the world on how you can achieve a better and more effective strategy. The multi-targeting ALL is currently a way, so instead of having a CAR-T which recognizes only one antigen, you have a CAR-T which comprises the capacity to kill and to recognize the two targets. Why? Because look at the number, the percentage in the right column. How frequent are basically the escape mechanism, which means failure of your treatment because the CAR-T is not active anymore because the leukemia has lost the antigen. And of course, there are many mechanisms which have been described. Can you believe that a CAR-T, so immunological intervention, can induce in the T cell, in the leukemia cell, a change in the genetic structure and coding sequence of the antigen which is recognized by the CAR-T, simply by inducing a pressure, an epigenetic pressure that induces a change in the genome. And according to the leukemia cells do not express anymore the CD19, which is recognized by your CAR-T. Amazing. Very impressive, at least for an old man as myself. The dual CAR is already a reality. This is the experience of our colleagues from Gosh in UK, that have already demonstrated by using dual CAR, you can achieve better remission and better control of the escape. I want just to briefly mention, and going to the end of my presentation, that in our institution, we took the challenge to say, can we address some of the critical issues that has been already highlighted by the current medical products which have been approved by the current regulatory authority. And we thought about that there would have been two major changes that we want to achieve. First of all, to use a non-viral approach, because still all of the current commercially available CAR are based on viral approach. And to do that, we use a non-viral approach. The second thing is that considering the fitness of the cell, because when you are taking from a patient the immune cells from the blood that has been under the pressure of the, let's say, chemotherapy, radiotherapy, the fitness of that immune system is not so high. And accordingly, we thought that it would have been a good idea to take a fresh lymphocyte from a donor. And so we decided to bring this to the clinic. It was a big challenge, because considering that you have to build up what is called the investigator medical products dossier to present to the regulatory authority is not very, and we are a very small center, not a big one. But we took the challenge because we believe that it is going to be a major breakthrough in the future for the treatment of cancer, not only pediatric cancer, but probably even adult, has been already shown for lymphoma. This viral approach work? Yes, and you can have exactly the same expansion as you can achieve by using commercially available products. And surprising to us, these cells can persist for a long time, thus controlling the disease. Of course, the question is not a biological question, it's a clinical one. You have to prove that these cells are equally effective as the ones that are currently commercially available. And this is the case. This is a mixture between adult and pediatric. Adult because for adult there is no access to CAR-T at the present time for relapsed refractory leukemia. So we are very happy about that. I don't think that it's going to substitute the commercially available, but it's a way to think about that when you have a breakthrough in medicine and that presents many challenges, the research is the only way to overcome that and to find a solution for the unmet clinical need. We are not alone because other colleagues around the world and even company, we are not a company, we are an academic structure. There are things about to provide by genetic manipulation lymphocyte from donor that could become an off-the-shelf products that can be used for all patients independent of the HLA. But of course, there are many other active research, and this is just to mention that many colleagues are working on different types of cells, not only T cells, but NK cells. I want to give the message that the breakthrough of the CAR-T, independent of the results that we did have so far, is a way of thinking the approach to cancer, which is not anymore just to kill through chemotherapy the cells, but to try to, in some way, to improve the effectiveness of our immune system. And we have already many examples. Think about the checkpoint inhibitors in adult solid cancer that, of course, has been shown to be highly effective, specifically in many types of cancer. Think about the history of metastatic melanoma, how there's been dramatically changed thanks to this use of these checkpoint inhibitors, which are basically another way to think about how to intervene with the immune system. Why academic research? Simply to explore novel concepts for the treatment of BCP. It's very important. Even if you have commercially available products, company might not be willing to pursue and to explore different approaches or different use of this type of breakthrough strategy. And this is the role of academia, trying to pursue a specific question, the use of novel treatment. Of course, still is not clear, and I believe that it's not the case, that you can consider CAR-T as a source of substitute to transplant in the setting of childhood LM, because a significant proportion of children who did receive CAR-T later on, either for the assessment of molecular remission or molecular residual disease, or the loss of B-cell aplasia, are basically enrolled to perform bone marrow transplantation. But again, let me share with you that 30% that I have shown in my personal experience, in our personal experience, and I have to say surprisingly that one of those patients is a Down syndrome patient, suggesting that even this fragile patient can achieve an impressive results for this type of approach. What does it tell you? That those patients can be kept for more than 3 years in remission without the need to perform another bone marrow transplantation. And what does it tell you? That this is possible. It's only a matter to understand why does it happen in one patient and not in the other. That might depend on the type of cells that you're using to transduce your CAR-T. or many other variables. But this is very important. You can achieve long-term remission. Emily Whitehead, maybe you can click to Google, is probably the most famous patient who had been treated firstly at UPenn 10 years ago. Relapsed refractory LM, 2 bone marrow transplantations she received. Now is after 10 years in remission, and she's still alive. So does it mean something that is feasible? And this is probably a strategy to pursue. Finally, let me just share how I treat relapse. Definitely, we do have standard chemotherapy and transplantation approaches. But I hope that I share with you what I believe is going to be the future for relapsed refractory ALL in children, because the paradigm in children with cancer is to cure more, but to cure better. Because better, it means to have the chance to provide a quality of life and the long-term free of side effect, which is equal mandatory as pediatric oncology. And that's, of course, completely changed our paradigm. Because, of course, when you had 40 or 50 years ago, the issue of fighting for life and death, there is no doubt. We have to do everything we can to provide life. But it's not anymore the paradigm, for at least for countries that can afford, by keeping in mind that we, of course, increase the percentage. And so these are basically a conclusion. Immunotherapy is changing the therapeutic scenario of childhood LL who relapse who are refractory. I have explained why Blina has been already in the part of the conditioning regimen process for transplantation for children who need it. I know it's going to be tested as comparison with the standard induction chemotherapy, and CAR-T are changing the therapeutic landscape of ALL and other lymphoid neoplasia, as you know, for non-Hodgkin lymphoma and myeloma in the adult setting, but it's still unknown whether it can substitute the need of transplantation in relapsed children ALL. And finally, let me thank all the people of the team from our place in Monza and from the University of Milano, but the teaching hospital is in Monza. And I'm very, again, pleased and honored to be with you today. And I thank you for your attention.
Thank you for the great talk. And I think we might have some questions. Of course. Oh,
yeah.
Sorry.
I think that there is a question, but probably you need the microphone. Maybe I can
Dr. Biondi, thank you very much for your presentation of your series of patients. There aspects of this disease. But there's was rated by clinical practice in the chemo-resistant
again. Thank you very much, Dr. Biondi, for your presentation about your series of patients. This is the immunological aspects of this disease, but there is very closely related biochemical pathways relapse of this disease and the chemo resistance and chemo-sensitive situation. If you explore some root and exosome-related microRNA silent pathways and considering this STAT3 interleukin 1 silent. And during the reprogramming of these genes between the chemo-sensitive and chemo-resistant stage situations. Therefore, they should be a checkpoint for this challenge.
Well, thank you very much, Emin. Of course, you open, I just focus on one aspect of the mechanism of relapse and accordingly on strategy that are currently exploring for overcoming the resistant refractory profile of a leukemia in a patient who relapse. And of course, as you said, there are many additional current strategy to overcome. And you mentioned the exosome, you mentioned pathway, which are basically being shown to be relevant for this type of profile. In my opinion, the innovation about the immunotherapy is that apparently seems to be independent by targeting with, of course, an immunological approach, the cells, independent of whether they are carrying specific alteration at the genetic level. So in that sense, in my opinion, it's novel, because if you take the CAR-T or the Blina or the INO, this type of, which basically target and kill the cells through the immunological mechanism, this seems independent by the mechanism which you can describe in the resistant cells. And in that sense, in my opinion, is novel, because the profile of acquired mutation and according genetic lesion are, of course, relevant for the type of drugs that you can use. But if you use a different way to kill the cells, and it seems independent, the success, because you're using a different mechanism. So that's, in my opinion, the immunological breakthrough seems to be as equally important as, of course, all the targeting gene approach that has been currently explored. So, but definitely, you're right. I just focus on the immunological approach and not all the other strategies that can be used. There is another question.
Thank you very much. Can you say something about CAR and T cells also?
CAR and K? Yes. I didn't have the time to get through, and just for the use of the different type of immunological cells to be transduced with the CAR is one of the very active area of research. For example, the type of cells that we are currently using are basically T cells, but polarized in a different manner as the ones are currently used in the commercially. Basically are called cytokine-induced killer cells. And these are basically T cells which express NK marker. The advantage of these cells, that even in different settings has been shown to have a very low incidence of GVHD as compared to the standard DLI or T cells that you can use. In the same way, NK cells are absolutely a very active area as a source, as alternative source. And in one of the slides that I didn't have the time to comment, our colleague from the MD Anderson reported last year in the New England Journal the use of cord blood derived NK cells. And we are currently pursuing as well the use of cord blood, because as I said, the fitness of the cells, of the T cells for this immunological approach can potentially make the difference. Because as I said, the T cells of a patient who have received many courses of chemo and radiotherapy is not the same as fresh normal cell from a donor. And in that sense, the use of either universal T cells or the use of alternative source to prepare the cells to be transduced with a CAR-T. And in that sense, of course, cord blood is a potential interest source we are pursuing as well. And could be in the future another alternative to have a product ready to be used. Because still one of the major problems is that you are dealing not with the drugs that you order to the pharmacy. So even in terms of planning the treatment of your patient, you need to be aware that you have a time for production, receiving, which in order to be optimistic is not less than 1 month, which is for relapsing refractory patient, probably too much.
Are there any questions from the audience?
Thank you very much for your nice talk. Maybe it's a little more detailed for the audience who are not pediatric hematologists, but I have a specific question about the blinatumomab use in relapse. So now I understand that the BFM also gives 2 cycles of blinatumomab for transplant, and Locatelli and colleagues reported 1 cycle. Did a big difference? Looks like BFM is convinced with the CUG study. I wonder, for example, if you have a patient who is MRD negative after first cycle of blinatumomab going into transplant, would you consider giving the second, considering the expenses of the drug? Thank you.
Well, you raise a very important question. First of all, as you have seen, the 2 studies in the conditioning in the treatment before transplant of the 2 studies were different. 2 cycles, 2 chemotherapy, 1 cycle. The point that has been at least by MEA and even in terms of reimbursement for us approved only what has been produced by the European study, and accordingly this is still the way we are treating relapsing patients. Might be that in the future this is going to change. Specifically to your question, because this is the real life. You have a patient, you are doing reinduction regimen with chemotherapy, might be still MRD positive or not so negative. You are giving 1 cycle of 29 days of blina. The child is happy because except for the first week or might be some side effects which occur not so frequently. At the end of the first cycle is negative. What are you doing? Is enough? This is very difficult to answer because apparently, at least for the very high risk patient, or maybe in high risk as well, the question is whether this is enough for achieving the results that you expect from transplantation, because you know the quality of the results of the after transplantation is the quality of remission that you have achieved before. Then of course you can argue what is the relevance of other components, but as you know, the quality of remission is essential. And you know very well that even in a patient in complete molecular remission before transplantation, you can still observe relapse. So I would be more conservative. Maybe it's a matter of age and white hair to say that I would prefer to stick on what is the still approved. So in a very practical manner, in our center, we are giving the cycle of chemotherapy, blina, and transplantation. But I'm always fighting with my transplanter colleague to say, why, Andrea, we should give enough? So in many cycles of chemotherapy, why not stop it and bring to transplant? Because you know better than myself than bringing a child not in a very good shape to transplantation. And actually, this is the challenge today for transplanter, as you know, because we are providing first line, second line of treatment, very heavy. And the complexity of transplantation is becoming higher. for complication, as you know.
I think your question is already answered, so I'll pass the thank you for...
Thank you very much indeed, again.
Dear distinguished guests and attendees, as being the first session of the first International Cancer and Life Symposium of Lösante, probably this is a historical session. I'd like to, in order to use the time more efficiently, I'd like to introduce you Dr. Dirk Reinhardt, who is very famous in the field of AML, and we already know him very well from the BFM AML study group and also from the international BFM AML committee. I was very surprised when I saw that he's also the director of the cardiology and pneumology departments of Ezer University as well. He's the director of UPAL, and from Üstün, Ezer University, Germany, Professor Dirk Reinhardt. We are very excited to have you in this meeting, and also we are very happy to hear about the latest advancements in childhood leukemia.
Yeah, thank you very much, dear colleagues. I really have to thank the organizers and especially the society to invite me for this very fantastic symposium. Thank you very much. We already have had yesterday evening a nice meeting. Oh, great. An honor to me to be here. You gave me a real challenge to speak after Andrea Bioni. Because I already anticipated that he will cover a lot of these late advancements in childhood leukemia. But as I'm a little bit more focused on acute myeloid leukemia, I guess I found some options to provide new information and data, maybe not only about therapies, but also about structures we will need and we think we have to address already now, but especially in the future. Let me start just a short overview. I would start with the structures and the relevance of international collaborations. This has been already mentioned with this long tradition of the so-called IBFM family, which have been founded, I guess, about 40 years ago. But we think that it's already time to even expand this collaboration and to put it on a formal level, especially in the acute myeloid leukemia, which is even more rare than lymphoblastic leukemia. And in the second part, I will try to touch some new therapies. This should just demonstrate, and these are some recent results from our panel NGS screening, how many genetically defined subtypes we already found in pediatric AML. And given that, for example, in Germany we have about 100 newly diagnosed AML, you can imagine these are already more than 50, 60 different genetically defined subgroups. One subgroup, 1 or 2 patients. Therefore, I guess it's a real need and a task to develop structures which might work on an even global level. And this initiative has been initiated by the Leukemia and Lymphoma Society from North America, together with the Childhood Oncology Group. A little bit later, we formed the so-called UPAL consortium in Europe, definitely to bring together all the stakeholders, institutions in Europe, including more than 27, 28 countries, to act as one counterpart to this US initiative. The idea is to identify and diagnose all relapsed and refractory acute myeloid leukemia children, performing a comprehensive characterization to get ideas about biomarkers, especially about the drug biomarkers and set up on a global scale sub-trials which address these specific biomarkers so that we, at least with the available drugs, can address each subgroup in a proper way. To set up this, we started with a registry, and here are some of the relevant institutions in Europe who already partners with the UPAL Foundation organization, just to set up not only these clinical trials, but also to set up these structures for diagnostics and for transfer of patients to the trial sites. This means that we definitely have to establish on a reliable and very professional level the diagnostics for pediatric AML in all kinds of options. It's still morphology. I'm a morphologist, I guess, yeah, it's a dying profession, but I still believe that morphology helps us to early and first identify children, especially in emergency cases like acute myelodysplastic leukemia or monoplastic leukemia. But for sure, we need all these actual methods and new methods, innovative methods of immunophenotyping, panel next generation sequencing, but also RNA-seq and as latest option, optical or genome mapping to substitute karyotyping in a more detailed way. What we did already within this UPAL consortium, set up a network of European reference laboratories. These labs have to fulfill highest criteria of quality because the data we would like to generate should be good enough to provide reliable and quality control data for market approval. It's not only about the diagnosis, it's only already to have these drugs finally licensed for children, which is not the case at the moment. Here, just an overview about the two networks, about the flow and the molecular genetics. One is driven by Michael Dvorak from Vienna, the other from Martina Pigazzi in Padua. We also set up associated to the WHO handbook for laboratory management and handbook, which just covers all the different issues of quality management and diagnostics in children. In a similar way, we also would like to learn more from all the available data which have been gathered in the last decades. So this is the Data Commons project driven by Sam Wolschenbaum from the University of Chicago, and the aim is to bring together all clinical and biological data from children with acute myeloid leukemia to be able to identify smaller subgroups, to characterize smaller subgroups, and even, if possible, to find better options for stratification. This has been executed in North America, Japan, Europe, and Australia, New Zealand. Just to give you an idea how this project is ongoing, this is a master protocol which just started in Europe and North America. And you may see we have on the left hand the screening. This is just to diagnose the children, to have the characteristics available. For example, is it a FLT3 ITD? Is there an MNL rearrangement maybe appropriated for an menin inhibitor? Is there the expression of CD123 to apply CD103 directed ADCs and so on? And this is the idea of how we would like to treat the children in this kind of sub-trials. Again, with a clear aim to finally improve these compounds in children. There are already approvals in adult AML. These are some new drugs. Some you are rather familiar with, like the Mylotarg, which is in this little, or in the same way extracted much earlier as the Immunology, which Andrea Biondi just mentioned. There are also rather conventional drugs already approved in the adult field, CPX351, which might be or could be the standard of care for children. There are FLT3 inhibitors which have been approved in the adult as well as IDH2 inhibitors. In all these subgroups, the final results in children are still lacking, but we hope that we finally can make these drugs available also in children. The same is true for the venetoclax as a BCL2 inhibitor. With this, I would just come to this overview. I hope in some way this cartoon already summarized a lot of these different options, and some of them have already been mentioned by Andrea Biondi. Here, the targeted antibody conjugated with the catecheramycin. We have another option for the CD123, which is a conjugation of this antibody to human IL-3 with the diphtheria toxin. For sure, the known FLT3 inhibitors. We have recent results also in phase 1 trials from these menin inhibitors, which might target MLL rearranged leukemias, but also NPM1 and NUP98 aberrations, and others like the IDH, like the MDM2 inhibitors, maybe not Idazanutlin, which is, I guess, already gone from the market because it was not successful in the adults, but there are still others which might play a role in the future. These are just an overview, and to extend this, and again, this also has already been covered in some way by Andrea Biondi. In ALL, we have these B-specific antibodies targeting CD19. We have the venetoclax. We have other monoclonal antibodies, but also, as mentioned, the CAR T cells. And with this, I will skip the blinatumomab. This has been already mentioned. Fortunately, I have only one slide. So this is done. And I also just want to mention with one or two slides the CAR T cell, because this is, as Andrea Biondi already mentioned, definitely the breakthrough in the last years, especially in the CD19 positive malignancies. It's much more difficult to imagine how we can use both the antibodies and the CAR T cells in myeloid leukemia, because you have not such an exclusive target as the CD19. With the CD19, for sure, you eradicate the B cells. This you can substitute just given immunoglobulins. In the myeloid field, you eradicate the whole myelopoiesis, and to be honest, this is not feasible, but or only feasible with a transplant as a secondary step. These are the original results from Shannon Moore about the CAR T cells. But let me go to the next step, and I'm very happy about the question you raised about the NK cells, because this is something I would like to focus on the next minutes. When we and others look for leukemia-associated targets in AML, for sure we know about the common expression of CD33. We know the common expression of CD123. We learned that CLL1 seems to be a marker which is specific for leukemic stem cells. And as shown here, these markers are definitely expressed on leukemic stem cells. However, others like CD38 are not. And therefore, there are therapeutic strategies in CAR T cells targeting these different antigens, but also B or 3 specific antibodies. What we addressed, and this have been mentioned in the answer from Andrea already, is definitely can we use also the NK cells in acute myeloid leukemia? The natural killer cells have an innate cytotoxicity against tumor cells, and they can recognize stress-induced ligands, so they seem to be effective. There are major advantages over the T cells, and this is definitely, as already mentioned, the off-the-shelf utilization. There is almost no GVHD. There seems to be much less cytokine release syndrome, and the neurotoxicity, which we also experience with CAR T cells, especially with blinatumomab, seems to be less frequent. It can be just get from healthy donors or even parents. So these NK cells might have some advantages. However, they are very active, at least in vitro. These are some results in our lab against different cell lines of myeloid leukemia. So they definitely look very active. However, if they are unmanipulated, a lot of tries to use it in clinics more or less failed. The clinical effectiveness of NK cells, just as NK cells, didn't really work. The trials already, the first one at St. Jude several years ago or 20 years ago, finally did not give a clear advantage of these natural killer cells if they are just applied as or not unmanipulated. It's mainly due to the pure in vivo expansion and persistence of these cells in patients, and this might cause a reduced clinical efficacy. However, due to the advantages, there are several other approaches to use natural killer cells. These are activated natural killer cells, as mentioned, like cytokine-induced natural killer cells, but also the support with interleukins, which might allow a longer persistence of the cells, but also the development to NK CAR cells. What are the mechanisms which finally prevent the effect of NK cells in the environment, in the microenvironment of the bone marrow associated to a myeloid blast? Indeed, there are several mechanisms by which the leukemic blasts dysregulate the activity of NK cells, but also of T cells, just by expressing different activators, by expressing different inhibitors and other molecules, which finally make these kind of cells less effective. So in the natural environment, the NK cells are not really active against the leukemic blast. This is a mechanism of escape and modulation of the microenvironment by the leukemic cells itself. And these are the different fields one can address. It's for sure the cytokine, external cytokine stimulation by IL-2, maybe also IL-15. However, the direct application of these drugs is very, very associated with a lot of side effects, so it's almost not possible or really difficult. For sure, this has also been mentioned by Andrea Biondi, there might be options to use checkpoint inhibition, maybe not the classic PD-L1, because these are not so much expressed on the myeloid leukemia, but TIM3 or others might be an option. There are data that the blasts secrete TGF-beta to inhibit the function of NK cells, so there are options to trap these kind of molecules to prevent the dysfunction of the immune cells. And then we come to the manipulating stuff. There could be an idea to have specific NK cell engagers, like the blinatumomab for the T cells, but also activations via CAR T-cell manipulation. And this is what I already mentioned. Yes, cytokines like the treatment with IL-2 or IL-15 improve the activity of the NK cells. However, they are very toxic if you just apply them as infusion. For this, our approach at the moment is that we build a CAR construction which allows IL-15 secretion and the secretion of the IL-15 receptor within the same CAR, and you can just select if you would like to use CD19, CD103 or CD93, and you see that all of these killer cells, at least in vitro, are very effective. And this is also true in vivo if you use PDX, patient-derived mice models. And this is just a demonstration how this works. This is just in CD19 positive cells, but as you may see, this is the CD19 CAR, and the effect is rather limited with the cells. However, if you just augment these NCAR cells with either IL-15 or the IL-15 receptor, the survival and the occurrence of leukemia is much less. So this might be a promising approach to overcome the limitations of the effectivity of NK cells in men. We would like to transfer this, as always, also into a clinical trial, so using activated NK cells, but also immune modulation for specific markers, and finally, such an NK CAR CD123 cell in a small group of patients. As Andrea Biondi mentioned, it's a hard work, maybe not to do this in the lab, in the mice, but to get all the approvals to get this done in the clinics according to GMP and all the results. So I hope with this I could add some other aspects to the already comprehensive talk from Andrea Biondi. And in summary, I would like to focus on this Leukemia Lymphoma Society panel UPAL initiative to establish a global database to allow subgroup analysis, as mentioned in the Interact consortium, to harmonize and improve the quality of diagnostics to a level which allows market approval, and for sure, which is the major aim to set up clinical trials, hopefully in the future much more biomarker-driven trials to get more children cured with less side effects. We heard about a lot of emerging therapies, targeted therapies, antibodies, small molecules, and this broad field of immune modulation, ADCs, but also the cellular therapies with CAR T cells, with CAR NK cells. It's still not known, or maybe it's, at least with the last option, necessary to, that this is just a bridge to transplant, because otherwise one could not imagine how to re-establish, at the moment at least, the myelopoiesis in children. So with this, I would like to thank you for your attention and thank all the people.
Dr. Reinhardt, thank you very much for your inspiring and excellent speech. I'm sure there could be some questions from the audience. So the field of AML is more gray at the moment, just not compared with the AML field. Because we have many many drugs at the moment that are currently in use, or for AML they are still in clinical trials and in the pipeline and also in the drug development stage. What's your guess about which would work much more? Do you think that it will be the ADC part or the CAR T-cell part for AML?
I guess, to be honest, the ADC part will be much easier. Yeah, because these ADCs have a limited half-time, and then you can go to transplant as soon as you achieve remission. Therefore, I guess this will be the easier way at the moment. However, it's again not clear how to finally apply that. The first clinical trials in adults, but also in children, the first approaches with the CD123, had major issues in terms of toxicities in the beginning. It's maybe an issue of dosages and relation between different cells, but all of these compounds have had at least some issues so far, and therefore it's hard to finally define which will be the best option. The CAR T cells, it's a bridge to transplant. Therefore the promise, which is true in some way, maybe not proven, but the promise that you can just use CAR T cells to cure the child is at the moment definitely not given in AML. Therefore it's a bridge, and then it's also an economic question, what is the better option?
Thank you very much. Are there any questions at the moment? Yes, that's great.
Dr. Thank you for your nice talk. And I would like to learn if you have an experience on tracheotomy in AML or JML as an MAK inhibitor. Tracheotomy.
Yeah. We have, but very limited, and unfortunately it didn't work. But maybe this is always the issue. There is the clinical trial is not focused on AML. And then finally you have one child, usually second, third relapse refractory, and therefore it's always hard to say is such an approach, well, it's failed due to 5 cases, or how we should decide on this. There's the same is, for example, the venetoclax. Very successful in adult, or especially in the elderly, combined with azacitidine or with others. In the children, we have already, there's one trial out, with combination with high-dose RRC from St. Jude Hospital, with a response rate of about, in total, 67%. But 67 is more or less the same we achieved with conventional chemo. So the question is, what is the addition of the venetoclax? I still believe there is an option for a subgroup, also in pediatric AML, but it's hard to define. And this is one of the trials we try to establish here, to find a place for each of these compounds, finally to have an optimized treatment. And to be honest, we will not find one treatment which fits all AML. That is a little bit the story what I showed in the beginning. We have so many genetically defined subgroups, and I'm afraid, or for researchers, it's a great field. We will need a treatment for each of these particular subgroups.
Thank you. And one more question.
And I would like to learn if immunotherapy can replace conventional chemotherapy in a near future.
I'm not, so it will not completely substitute conventional chemotherapy, because we already, and I will show this in the later, I think we already also achieved very good results in AML with conventional therapy. So it's a long way to substitute. But I guess, as also Andrea mentioned, there are several steps, or parts of the treatment where we can use this. This is conditioning regimens, and what seems to be very promising at the moment is post-transplant immunomodulation. For example, what I mentioned, the combination of an MDM2 inhibitor with donor lymphocytes seems, at least in adults, very promising as a post-transplant. We have a study from Frankfurt with cytokine-induced cells post-transplant in those children where one has also post-transplants, the risk of relapse is very, very high. This again showed rather promising results. So there will be parts of the treatment which will definitely will be substituted or even improved by these immunotherapies.
So I guess there are no more questions. Thank you very much, Professor Reinhardt, for this nice speech and also for your continuous support to our patients. Because we are writing you some emails about some BFM patient questions. So thank you very much for your continuous support.